Today, we are going to tackle some really important questions, especially one of the key command words you will see again and again in your exam papers. And I thought from the thumbnail you might have already guessed it. Yes, it's all about discuss. Now, I know many of my students, and maybe you too, feel that discuss questions can be a bit disgusting. The truth is, you already know the concepts, but the challenge is how to write them in the right way to score that full 6 out of 6 marks. I'm not sure how many of you have noticed this. If you look at your textbooks in the last pages, they have described the command words and what exactly a student needs to write. Based on that, this is what they have given for this case. So, according to this, I have made some easy steps for you to write the answer in a proper way. The steps are so simple. It's spot, look, tell, analyze. And today we are going to learn these steps with a practical example. This is a question from the October 2021 6C paper. I specifically choose this one because when I was preparing questions on chromatography, I came across this essay type question. That's why I picked it. But remember, you can apply this method to any discuss type question. So, first step, you have to read the question very carefully. I'll give you 1 minute. You can pause the video and read the facts they have given. The question is, discuss the effectiveness of these extraction methods in the identification of these species. Use the information in the graphs to support your answer. Now, let me explain this question in a very simple way. They have 4 different species of plants, basically seaweeds. You can think of them as sea plants. What they did was they took 2 equal samples from each species. One sample was tested with propanol and the other with diameso. Then they separated the pigments chlorophyll A, chlorophyll B, and total chlorophyll. After that, they measured the concentration. So, this is the question we are going to work on today. Now, let's look at it step by step. So, the first step is the most important one spot. This is actually the step where most students fail because they don't even spend 1 minute to spot the question first. Please remember, reading and spotting are 2 different things. Now, in this question, the main part is effectiveness of these extraction methods in identification of species. That's where I'm going to spot. Spend at least 1 to 2 minutes on this process. Even though the question is clearly written, you can still underline the keywords. This underlining method helps you stay coordinated because in essay type questions you have to write a lot. And while writing continuously, you can easily lose track and start writing something else. If that happens, you will lose marks. And that's why underlining is so important. So here I will underline the words effectiveness, extraction methods, identification of species. Now, when I spot these keywords, I know I have to analyze the effectiveness, check how good these methods are, and how they can be used in identifying species. The whole question is built around that idea, using these methods to identify these species. Here is one more tip. While practicing, you can write some sub questions. This is a very good practice, especially 1 month before your exams. But remember, in your final exam paper, don't waste time writing sub questions. This is only for practice. Why? Because when you write sub questions, you are training your brain to think more deeply about the main questions. You are analyzing it from different angles, and this will help you to understand the question better. Later, during the real exam, your brain will automatically start analyzing without you needing to write them down. So now pause the video and try to write some sub questions. Here you are not a student, you are the examiner. Imagine you are setting this exam paper if the main topic is this. What smaller questions would you create? I'll share a few examples that I wrote. Which method works best for each species? Can we use this method in real life? What problems might happen with this method? Are some methods better for some species than others? Why does a different solvent extract a different amount of chlorophyll? These are the kind of sub questions I came up with. The next most important step is look. At this stage, I ask my students to look like a scientist. That means you are not just looking, you are looking with questions in mind, trying to discover something. Most of these types of discussion questions are given with charts, tables, or graphs. So, when you're looking at a graph, you need to focus on things like titles and axes. This is where many students make mistakes. Now, let's look at the example. Let me show you. In graph 1, the title shows the concentration of chlorophyll A in 4 species extracted using 2 different solvents. In graph 2, you can see the concentration of chlorophyll B in the same 4 species with different solvents. And finally, in graph 3, the total chlorophyll concentration in all 4 species is shown. And when you look at graphs, you can mark important parts like circling the x's with a pencil or pen. This might help you to pay attention to the details. Now, when analyzing, you must compare the heights of the bars almost like solving a puzzle. Ask yourself which bar is taller, which one is shorter. That's how you begin comparisons. For example, here in graph 1, chlorophyll A is highest in species P with propanol. Also, species P, R, and S have more chlorophyll A with propanol than with DMSO. But there is an exception in species Q, DMSO extract more chlorophyll A than propanol. And if you look at graph 2, chlorophyll B is highest in species Q with propanol. Also, species P and Q show higher values with propanol than DMSO. But again, there is an exception. In species R, more chlorophyll B is extracted by DMSO than propanol. In species S, almost both solvents give equal results. And when you look at graph 3, the total chlorophyll concentration, we see that propanol generally extracts more total chlorophyll, especially in species P. So you can conclude overall propanol is the more effective solvent while the MSO is less effective. And also, if you compare graph 1, graph 2, and graph 3, again you can see some inconsistencies when the chlorophyll extracted alone versus when they are extracted together. So, from this, you can also spot patterns in the graphs. For example, ask, is one solvent always more effective or does it vary depending on the species? And also, don't forget to notice exceptions because examiners love to test you on those. For instance, in graph 1, species Q is the exception where DMSO is more effective than propanol. And the next most important step is tell. So tell what we are going to tell. Comparison and general trend. So here, whatever you have spotted and whatever you have looked at, you are going to write it in your exam paper. Basically, we can divide it into two, comparison and general trend. And within our example, we can see comparison, right? So I can see more chlorophyll B in species Q with propanol and also more chlorophyll A in species P, R, and S with propanol than with DMSO. So these are some comparisons, and overall, I can say the general trend is that propanol is more effective and DMSO is less effective. So this is the general trend which I derived from the comparison. And the final important step is analyze. Now, analyze is super important because in this type of question, most of the marks actually come from the analysis part. So, what do we analyze? Basically, it's where you apply some high level thinking to the question. If I look at this example, right, here is my first analysis. So, we have already spotted everything, we have written it down, and now comes the thinking part. The idea is to check how much this extraction method can actually help us identify the organisms, or in this case, the seaweeds later on. So that's the whole point of the question. You have done the experiment in the lab, and later they ask, by extracting chlorophyll, can you identify the seaweed exactly? That's the key. Now, here is what I have noticed. There is an inconsistent difference in results when pigments are extracted separately versus when they are extracted together. If you look at the graphs, you will see this. Sometimes, when extracted together, some chlorophyll might be lost, so the total ends up less than the sum of the individual pigments. On the other hand, in some cases, the totals are actually higher when extracted together more than the sum of the individual values. That could mean that along with chlorophyll A and B, some other pigments are also getting extracted, which explains the inconsistency. So, this is one of my important analyses. Basically, these variations create inconsistencies, and that can make species identification tricky, especially if different methods are used interchangeably. For example, if a scientist in one country analyzes pigments one by one, he may not be able to properly identify the species because of these inconsistencies. Next important analysis is that the choice of solvent depends both on the type of chlorophyll and the species being extracted. Since different pigments vary in solubility and membrane permeability across solvents, no single solvent can be universally applied. From the graph, we can see that in species P, R, and S, chlorophyll A is extracted more effectively with propanol, yielding a higher concentration. However, in species Q, the trend is different. DMSO produces a higher extraction yield than propanol. But this applies only to chlorophyll A concentration. The next point to analyze is the use of multiple solvents for species identification. From the variations we observed, it's clear that a single solvent may not extract all pigments equally well across different species. Different solvents yield different concentration of chlorophylls depending on the species. Therefore, using multiple solvents can enhance the accuracy and reliability of the species identification. To do this identification, we should have some pigment profiles already, as shown in this picture. This kind of comparison tables or calibration curves is needed to identify species by their pigment profiles across various species. And these tables and curves should include the specific pigments along with the different solvents used for each species. Another important point is that this method avoids the complicated and costly process of DNA analysis. Compared to DNA based techniques, chlorophyll extraction is a much simpler test method. It can be performed easily even in basic lab settings or directly in the field. According to the graphs, propanone can be used as a single solvent for species identification since it shows greater variation in pigment extraction between species. And this variation makes propanone effective for distinguishing species using only one solvent. Another important point is that whenever you are given a graph or a data set, you should always check for statistical parameters such as the mean, standard deviation, and error bars. However, if you look at this graph, none of these parameters are included. This means that while you can still analyze and interpret the given data, you cannot be confident about the variability or reliability of the results. Therefore, it's essential to highlight this limitation. If statistical parameters are provided, you should point them out. And if they are missing, you must also emphasize that this information is absent from the data.